Colloquium series. Glad to see you. This is going to be our first talk of the series for this year, and we're really honored to have Dr. Greg Miller here. If you're wondering about the seminar series, we're really excited about who's coming this year. We have, I think, 7 speakers this semester, 6 speakers in the spring, and then it's not related to the Colloquium series, but we do have a research day event that's also listed on the flyer at the end of April. We have a lot of really nice big name speakers coming in presenting really interesting research.
So we are very pleased to have Greg Miller here today to kick us off for the Colloquium. And we're going to invite Idan Shalev from BBH to make the introduction.
So let me first of all just take this opportunity to thank Chris England and Anne-Marie Chan for doing a great job and for organizing another fantastic colloquium this year. Amazing, fantastic line of speakers. So maybe we can just give them a round of applause for their efforts. And what it starts for the year with Dr. Greg Miller. Sorry for raising the bar. So Dr. Miller is a professor. So first of all, I'm happy to introduce to you my friend and colleague, Greg Miller. Dr. Miller is a professor in the Department of Psychology at Northwestern University, and he's also affiliated with the Departments of Medical Social Sciences and the Institute of Policy Research, which can give you some hint to the interdisciplinary breadth of his research and the translational aspect of his work. Greg received his Ph.D. in clinical psychology at UCLA and completed a clinical internship at Western Psychiatric Institute in Clinic, which was followed by a postdoctoral fellowship in health psychology at Carnegie Mellon University, where he worked with Dr. Charlie Cohen, which was one of the colloquium speakers last year here. Greg joined in 2000 to the faculty of Washington University in St. Louis. And after 3 years, he accepted a position at the University of British Columbia, where he was a professor of psychology before he moved to Northwestern in 2012. Together with his lifelong collaborator and partner, Dr. Ruth Chen, he co-directed the Foundation Health Research Center at Northwestern, and his research examines the behavioral and biological mechanisms through which stress affects health. It's quite relevant to the work that we're doing here in the college. His lab brings together theories and methods from across the behavioral and biomedical sciences in order to understand the stress-health connection, and you will hear a lot more on his research soon. Overall, his extent research is really influenced many in the field, including my own work. His work is supported by grants from the NHLBI, NSCHD, and NIDA. Previous studies has been supported by the Brain and Behavioral Research Foundation, previously known as NARSOG, the Canadian Institute of Health Research, the Michael Smith Foundation, the Heart and Stroke Foundation of Canada, and the American Heart Association. Dr. Miller has received a number of honors for his research, and we'll just name a few of those. The Young Investigator Award from the Society of Behavioral Medicine, the Early Career Award from the American Psychosomatic Society, and the Distinguished Scientific Award for Early Career Contribution from the American Psychological Association. It's quite an honor to receive. He is currently President-elect of the Academy of Behavioral Medicine Research. He was Associate Editor of the Journal of Psychological Bulletin and Psychosomatic Medicine. And he's currently a consulting editor in both of these journals and in the journals of Brain, Behavior, and Immunity and Clinical Psychological Science. And just finally, together with Dr. Chen, Greg has also mentored many students who went on to become successful scientists, securing faculty position, among them our own Hannah Fryer. So today is going to talk about the health consequences of socioeconomic disadvantage in their early years of life. So let's give him a warm welcome.
Thanks for the great introduction. Can you hear me back there? All right. So it's really fantastic to be here and get a chance to see old friends and to make new friends. You guys have really such a fantastic group of scholars here coming at things from so many different angles. So it's really been a great learning experience for me. Just 8 hours or so I've been here today. So let's continue that. I've got some slides, as you'd expect, but I'm not committed to getting through them or So jump in with questions or with conversations or with whatever you want to say. I'm happy to make this more interactive and less formal if you want. I'll just go ahead and get started here. So we've known for many, many years, and in many ways thanks to people at this institution and in this room, about the life trajectories of kids who grow up in economically disadvantaged circumstances. And we've known for a long time that they're at greater risk for, on the whole, worse outcomes in academic and employment settings, for worse psychiatric outcomes, for more encounters with the juvenile justice system. And all those findings have been demonstrated elegantly in intervention work. What's been surprising in that literature, though, probably over the last decade or so, is the increasing evidence that early life adversity, particularly in the form of low socioeconomic status, not only forecasts challenges and vulnerabilities in the educational and behavioral and psychiatric domains, but also seems to relate to people's risk for chronic diseases associated with aging that typically arise when people hit their 50s and 60s and 70s. And that's been a real revelation. So I could show you lots of different studies depicting this association. Here's one of the ones that I think was early and compelling. So this is a study of about 5,000 Britons who were born in the post-World War II period. It's a prospective epidemiologic study that began in the early part of the 20th century, the middle of the 20th century, sorry about that. And they crudely stratified the participants in the study according to their father's occupation, whether it was manual or non-manual. So this is a very crude measure of childhood socioeconomic conditions. And at this point in the study, it followed up until the mid-50s. And what you see is that the mortality risks for these two groups of crudely defined subjects start to diverge when they're in their early 40s, and they keep diverging, so that by the time they get to their mid-50s, there is about a twofold difference in rates of premature mortality, favoring those who come from more advantaged childhoods. And these differences are independent of traditional risk factors for premature mortality. So you can throw the relevant covariates in the equation, and most of them are prospectively measured. And this difference persists. You might say, well, you know, it's a twofold difference in early mortality, but you're only talking about 6% versus 2.5 or 3%. You know, does that really matter? Of course, when people are in their mid-50s, they're not dying very often. If you follow people out until their 70s, 80s, 90s, when rates of mortality are much higher, you still see these curves separate about the same space. So this is a fairly reliable finding in the literature. Early childhood socioeconomic status predicts premature mortality above and beyond conventional risk factors for mortality, including adult socioeconomic conditions. So there have been a few dozen studies that have shown this, many looking at all-cause mortality, many looking at cause-specific morbidity and mortality. A lot of the best evidence is from cardiovascular disease studies, just because there's the best epidemiology there. There's also been some good work, not just on the length of life, but on the quality of life. So these are data from Mark Hayward, who's one of your former colleagues here at Penn State, bumped into on the plane yesterday coming here. And so Mark's done some fantastic work in the context of the health and retirement study, using 4 or 5 ways of data to characterize deterioration in people's functioning as they reach older ages. And what he shows is that childhood socioeconomic status in this cohort and in others is a robust predictor of how quickly people are going to deteriorate and how many active years of life they're going to have. So in this particular slide, you can see this is the number of childhood socioeconomic adversities from none all the way to 5+. And on this axis, you're looking at active life expectancy up to age 50. And then what he's done here is stratify it by how much education people obtain. And you can see that within each of these educational categories, college, some college, high school, less than high school, there's a gradient according to childhood socioeconomic status, such that those who come from more advantaged conditions have more active years of life post age 50. So you can't completely get rid of this even when people get high levels of education, although getting higher levels of education certainly does increase your life expectancy, your active life expectancy. So there are a lot of studies showing this. It's pretty robust. The measures of childhood SES are sometimes kind of crude, sometimes a little bit more refined, sometimes really good, like in Mark's work. And now there's starting to be some evidence that these associations between childhood conditions and health later in life not only extend to things like heart disease, disability, premature mortality, but there's some hints that they might actually go into the next generation, so that my socioeconomic conditions as a kid might have some repercussions for my own children's health. These are some data that we've produced looking at a mother's childhood socioeconomic conditions and risk for adverse pregnancy outcomes when she goes to deliver. So you can see here as we're looking at preterm birth, so this is delivery at 37 weeks of gestation or shorter. This is small for gestational age, so less than 10 percentile sex and GA-adjusted weight, so with growth restriction. You can see that these pattern by maternal socioeconomic conditions, maternal childhood socioeconomic conditions, and these are independent of mother's socioeconomic conditions during pregnancy. So again, this is a childhood effect, not a concurrent effect. As best you can separate those things statistically, and again, controlling for most conventional risk factors for adverse pregnancy outcomes. There are other data like this in the context of heart disease risk factors and in asthma. We have other data suggesting that there might be cross-generational influences. Again, the mechanisms that would cause these associations to arise are really unclear, and this is a pretty early area of research. You can't obviously do tight randomized studies, but they do converge quite nicely with animal models showing cross-generational effects. So there's some thinking that they might in fact be causal, but we've got a long way to go in this area. So why should we all care about this? I don't think in this crowd I have to argue that much about this, but certainly when I talk in traditional psychology departments, people say socioeconomic status, why would you study that? We can study right-handed undergraduates all day long. So obviously these findings have implications for families, they have implications for public health in this nation and others, implications for economic productivity. If these really are causal effects where the childhood circumstances that we're raised in have repercussions for our health across the lifespan, maybe for the next generation, then if they're causal, then we're going to be feeling the repercussions of today's socioeconomic environment for many generations to come, right? The Great Recession that occurred between, say, 2008 and 2012 really changed the economic landscape for lots of families in our country. And if those are sensitive periods for lifelong health, then 50, 60, 70 years down the road, we may be feeling that. I think from a purely kind of scientific perspective, these sorts of observations pose real challenges, conceptual challenges and mechanistic problems that people working in interdisciplinary settings like this like to take on and are hard but tractable problems. So, you know, the first thing you need to answer when you're thinking about how these associations would arise is there a plausible behavioral and biological pathway by which you get from broader socioeconomic conditions in childhood down to the level of how tissues function and how cells function in those tissues and gene expression patterns and all other metabolic processes get affected. So you need to connect the dots between the larger social context and molecular biology, and that's hard to do. You know, we have from decades of stress physiology work in animals and humans some idea of how that happens, but the details are still pretty sketchy. I think what's harder here, particularly for people like me who come from more of a background in stress research, is dealing with the temporal aspects of this. So the fact that the exposures we're talking about, which happen during the first 5 or 10 years of life, are separated in time from the outcomes we care about, risk for heart disease, premature mortality, often by 5, 6, 7 decades. So you've got a really long incubation period between the relevant exposure and the relevant outcome. And stress research in general is quite good at describing what happens in the moment when people are feeling stressed. So, you know, I'm up here standing, lecturing, people are giving me kind of dazed looks, my HPA axis is activated, there's more cortisol going around. We're good at these sorts of things. But our models aren't very good at explaining how you can bridge a 50-year incubation problem, right? So how do you take an exposure like low SES, like maltreatment, and propagate it not only across biological systems, but across years and across decades in a way that is going to contribute to diseases that don't at least manifest clinically for quite some time. And developmental scientists have been really good at thinking about those sorts of issues with psychosocial outcomes at the end. Health psychologists really haven't, and stress physiologists haven't. So this kind of work requires, I think, both groups coming together and sharing their insights. So that's been gratifying but challenging. And then, of course, I've talked about risk up until now, but obviously there's a lot of variability in outcomes for kids coming from socioeconomically disadvantaged circumstances. And, more than half the battle is explaining why it is that some kids do great and are resilient or protected, and be able to identify the strengths that they bring to the situation and figure out how we might be able to harness them to help other kids and families do it in these circumstances. So that's another big area of inquiry that's important both practically and challenging scientifically. So when people see these data on childhood SES and later health, kind of two explanations often come to mind pretty quickly, right? So one of them is it's all about lousy genes, right? And the argument gets formulated in a couple of different ways, but usually it comes down to something like there is a basket of risky genes, and that basket of risky genes travels in families, and it predisposes families to do badly in school and have bad health problems, and everything that goes wrong is because of some basket of genes that travels in families. So what you're really seeing is not a causal effect of early life adversity on health, but some residual confounding by genes. Now, it's certainly a plausible explanation. It's kind of hard to test, right? Because usually those arguments don't come with hypotheses about what the relevant risk alleles are. If somebody had an idea that we could measure them, test it. So what can I tell you? I can tell you one, you can look at animal models, and animal models aren't perfect representations of childhood hardship. But in rodent models and non-human primates, you can do some pretty good approximations of those sorts of things. And when you do those studies experimentally, you see causal effects of early conditions on later risk for heart disease and stroke and other health problems. So those animal models, I think, are a pretty good basis for thinking that some of what we're seeing is causal. You can also look at administrative databases from Northern Europe, where you can study through tax records and medical records kids who were adopted in the first few years of life out of high SES circumstances into low ones, or more commonly the reverse. And those adoption studies are nice natural experiments, not perfect, but as close as we're going to get in humans. And in those studies, what you see is that the SES of the adoptive family relates to future health outcomes just as strongly as the SES of the biological family. So both matter. So that argument, those data would suggest that yes, genes are part of the story, but they're not the only part of the story. Context matters. So the other explanation people often arrive at when they see these data is that it's really not a childhood effect that you're talking about. That in countries like the United States, there's not as much social mobility as we'd like, and the childhood socioeconomic conditions are really just a proxy for people's adult socioeconomic conditions, and those are much more proximal in time to things like getting heart disease and stroke. And so what we're really measuring here is just using childhood SES as a proxy for how much education people are going to get, what kinds of lifetime wealth they're going to accumulate. That doesn't turn out to be the whole story. As I mentioned, in some of those epidemiologic studies, there are statistical controls for adult SES that are usually pretty well measured. The associations almost always survive that. You can also look at studies of mobile groups. So these are data from the precursor study. This is a cohort study done at Hopkins Medical School, where they took male physicians who graduated in the 50s and 60s when Hopkins was only taking males, and they followed them and looked at health risks. And you can see, so these are all physicians trained at Hopkins, right? As high in SES as you can get in the United States. They're physicians, they've been trained at prestigious medical school, lots of education, lots of income. You can crudely divide these physicians according to their childhood SES. Again, did dad have a manual versus non-manual occupation? And even in this very high status group by any metric, you still see heart disease risk in this case patterned by childhood SES independent of traditional risk factors. So it suggests that there's some stickiness of early experience. that isn't just captured by where people end up later on in terms of education and occupation. All right, so how then do we connect early experience with later health? Lots of different pathways. This isn't a simple story. This isn't a classic kind of 2x2 psychology, ABC. We and others have proposed models like this that involve all the different exposures that in our country travel with socioeconomic disadvantage. Some of the stories certainly involve the social environment and differences in family stability and family relationships and household conditions. And some of it involves physical exposures and differences in exposure to pollutants, intoxicants, and irritants that also co-vary with socioeconomic status in many communities. We'd argue that some of it goes through brain development that affects the way people experience the world, the social relationships they have, their lifestyles. Some of it is more of a classic programming effect. These are the kinds of questions that Don asked about. And we think that it's bold, that there's probably some early life programming of certain biological systems, certain parts of the brain, certain parts of the immune system where you get kind of a classic fetal origins kind of effect. But that's not the whole story, right? That there are continuing and sustaining and mitigating things that unfold across later childhood, across adolescence, and well into adulthood that can either accentuate those programming effects or offset them. And so we think that these kind of life course stories and these programming stories are pretty compatible, actually, even though the literature tends to be divided into people who think it's all about what happens in the first few years versus people who think that there's continuing plasticity. It seems to me that this is kind of like the nature-nurture debate. It's crazy to think that it would only be one or the other. From an evolutionary perspective, it seems kind of crazy to think that you would hedge your bets on programming every system in the first few years of life when humans typically live much longer than a few years. You can't make those kinds of forecasts. On the other hand, we know from really tight animal model studies and human studies that there are quite sensitive periods where things do get locked up. So you have to have a theory that accommodates both. Our focus has been on inflammation. And you saw that in the last slide. So we've been very interested in the idea that what early life adversity around low SES, but also around maltreatment and other exposures does is change the way that certain cells in the immune system respond to infectious and non-infectious stimuli, and that in shaping the way that those cells behave, you set up what we think of as a pro-inflammatory phenotype that carries across the life course and accentuates, along with genetics and other exposures, people's risks for a variety of different health problems. So we study inflammation because we think it's a pathway to many, many different mental and physical health problems that all pattern by SES and all pattern by early experience. And this is just a figure from a Cell paper showing all the different ways that non-resolving inflammation is thought to contribute to pathogenesis of things ranging from atherosclerosis, where a lot of our work is, to classic autoimmune conditions, to asthma, MS, some forms of cancer. So that's the main reason for our focus on inflammation. Also, we can get a hold of the key cells, so it makes it a little easier to do the biology when you have the cells you want. And our kind of overarching framework has been this very simplistic idea that what early conditions do is shape the way that certain cells, known as monocytes in their immature form and macrophages in their more mature form, respond to stimuli. And that these cells have a ton of phenotypic plasticity, they're quite responsive to their environment. And our argument, and you'll see some of the data on this later, is that early disadvantage kind of calibrates these cells to be more responsive to threats coming from pathogen-associated molecular patterns, and these are classic common bacterial and viral motifs that cells encounter a lot, and then danger-associated molecular patterns. These are stress signals from cells that are dying or injured. And that what early adversity does is kind of make monocytes and macrophages respond to those threats in a more aggressive way, increase their cytokine production, increase their homing to damaged tissues. At the same time, the cells become a little bit less sensitive to signals that normally are involved in regulating that process, so they become insensitive, or relatively insensitive to glucocorticoids in particular. And then as a result of that kind of calibration or tuning process, kids who grow up in more disadvantaged circumstances have more of this chronic low-grade inflammation happening systemic. And then across the life course, that has consequences. But this process isn't in itself pathogenic, isn't in itself adaptive or maladaptive. In fact, one could make arguments that having stronger monocyte and macrophage responses and less sensitivity to addition would be quite adaptive in the context of certain viral infections, bacterial infections, that as a kid you're going to be sick or less. We think that could actually be quite true. Our concern has been more what happens at the other end of the life course with chronic diseases of aging, where ongoing inflammation is the problem. So this very well may be kind of a trade-off where it's adaptive and protective in the early years, but as you go on, the costs catch up. So just a little primer on inflammation for those of you who don't do this work. I know it's a very interdisciplinary department. This is a monocyte. One of the main receptors that's expressed on the surface of monocytes is TLR4, toll-like receptor 4. You don't need to know that in detail. What TLR4 does is recognize this little red molecule here called lipopolysaccharide. That's a molecule that sits on the surface of gram-negative bacteria. And when the monocyte recognizes LPS, it starts the signaling cascade, and the details of that aren't important. What you need to remember for the talk here is that the signaling cascade culminates in the activation of this molecule right here called NF-kappa B, or nuclear factor kappa B. This is the primary pro-inflammatory transcription factor in these cells. And what it does when it's activated is scoot into the nucleus, switch on genes, and those genes code for interleukins and other cytokines and chemokines and mediators that allow the inflammatory response to go forward. And so what we're going to do in a lot of the studies I'll show you is we'll take blood from kids and adults. In vitro, we'll challenge the cells with LPS. We'll look at how well the cells produce these cytokines. We'll do some gene expression studies so we can measure activation of these processes here. And that's kind of the rev-up process that we hypothesize about, the increased reactivity. We'll also try to model a little bit of the sensitivity to inhibitory signals. So one of the main endogenous systemic responses to inflammation is activation of the HPA axis. So these cytokines right here head up to the central nervous system where they signal the HPA axis to get activated, and you get cortisol release. Cortisol, look at that. And that's high tech. Cortisol crosses the cell membrane, it binds to glucocorticoid receptors, which are sitting around in the cytoplasm. And then what that complex does is block NF-kappa B. It tethers up NF-kappa B so it can't go switch on pro-inflammatory genes. So that's why when you have a rash, you've got an autoimmune disease, you often take synthetic preparations of cortisol topically or orally because it ties up this signaling pathway that leads to inflammation. So we're going to do some in vitro studies where we feed cells cortisol at the same time we give them LPS and look at how well it blocks this pathway. So now we'll get to some data. So our first study on this was about a decade ago. It was a fairly small pilot study where we just wanted to test out some of these hypotheses in a preliminary way. We recruited a small sample of healthy young adults. They were between the ages of, say, 25 and 45. And we recruited them to be either high or low in childhood socioeconomic status, which was defined according to their parents' occupation. We case matched them on current socioeconomic status to take that off the table as a potential compound. So what I'm showing you here are differences between subjects who came from higher or lower early life SES conditions. These are adjusted for current SES by design, for other demographic and biobehavioral factors adjusted statistically. And what you can see here is when we target toll-like receptor 3 with poly-IC, which is a viral analog, it's making the cells think they're seeing a double-stranded RNA virus. The cells produce more interleukin-6 in culture if the subject had an early life low SES family. And then if we target cells with flagellin, which is a bacterial product that acts through TLR5, we see the same thing. We've looked at all the different toll-like studies, so most viral and bacterial and microbial endocrine stimuli, we see increased cytokine production in those from more disadvantaged childhood circumstances. It's not all of them, it's not always the case, but in most circumstances with most of these pathways, I won't go through them one by one, we see a pretty consistent effect. Is it biologically significant? Is it big enough to make a difference in disease? Nobody knows the answer to that question. I mean, we're talking here about, a 30 to 40% in some studies a 10 or 20% difference in vitro cytokine production. Neither we nor anybody else at this point has the epidemiology to know whether, when you magnify that against all the exposures that kids have across the life course, whether it results in differential diseases. We'll know more in 5 years about that. But we see this consistent pattern that is similar to what the model proposes, which is that growing up in more disadvantaged circumstances seems to be associated with the tuning of these particular cells towards more aggressive in vitro responses, at least. We did a bunch of gene expression profiling in these cells to look at patterns of gene network activity. And so one of the things you find, these are quiescent cells that haven't been activated or stimulated, is that even at rest, there's an upregulation of genes with response elements for nuclear factor kappa B. You'll remember that this is the main pro-inflammatory transcription factor, right? This is the one that slips in the nucleus and switches on pro-inflammatory genes. So even at rest, there's an upregulation of genes with response elements for that transcription factor in those who come from more disadvantaged circumstances. There's a downregulation of genes with response elements for the glucocorticoid receptor. So it means that in our young adults who came from more disadvantaged circumstances, genes that are supposed to be switched on by glucocorticoids by the HK axis are less switched on than they should be, right? So that's consistent with this idea that I told you about 5 or so slides ago, that there's less sensitivity to glucocorticoids. We measure glucocorticoids in these subjects. We have them collect gallons and gallons of spit. We can measure glucocorticoid receptor protein or messenger RNA. There are no differences at all in circulating cortisol, in salivary cortisol, in receptor expression. This is all kind of a genomic effect that's occurring at the level of the signaling pathway. There's something happening between the time that cortisol molecule crosses the cell membrane and gets into the cytosol and the time it gets to the DNA. That's different for people who come from different early childhood experiences. It's way beyond my knowledge to tell you exactly what's happening there, but you consistently see this. So those of you who are developmentalists will be saying, okay, so you're studying early life exposures and relating them to people in their 20s and 30s. How early does this happen? What's the developmental course of this? We did some studies in Manitoba looking at this question. So this is a study of 9-year-olds from whom we drew blood and did some of these in vitro experiments that I was showing you about. And we modeled the 9-year-old's life course, SES, all 9 years of it. And we can show through some cluster analytic techniques that in Manitoba, you've got stably low SES families, you've got stably high SES families, you've got families that are mid and mid-high, and then families who SES increases as the kid gets older. And the kids' IL-6 responses to LPS patterns along those lines. So the kids who come from the most stably disadvantaged circumstances produce the most interleukin-6 in vitro in response to LPS, and the kids who come from the most advantaged circumstances produce the least, and the ones who have increasing SES mobility will look just like the ones who have been stably high, suggesting that this may be about kind of the recency of their experience as opposed to what happened earlier. So we see this in 9-year-olds. We have been figuring out ways to get younger than 9, because getting blood from 4 and 5-year-olds is always a challenge unless there's a clinical indication. So more recently we started doing pregnancy studies, because we can get as much blood and tissue as we want. This is some really hot off the press data where we've been doing a lot of work in obstetrics the last couple of years, and we've been collecting placentas at the time of delivery to try and understand differences in placental inflammation and gene expression patterns as a function of maternal SES. So in our first study, what we did is collect the placentas and simply hand them off to a perinatal pathologist collaborator of ours who did a lot of staining for acute and chronic inflammatory regions. So these are chronic inflammatory regions. So we just code people here in discrete analysis as is there an inflammatory lesion present in any of the three major placental compartments? It doesn't vary by SES, and as you can see, it varies really linearly with SES, such that the highest rates of chronic placental inflammatory lesions come in the women who have the lowest income category, and it goes down unless there's dose response fashion here. Most of these lesions are either happening on the maternal side, so in the decidua, for those of you who are placenta pathologists, or they're happening in the villi, which is the innermost layer where nutrient exchange between the mother and fetus occurs. So these are a little bit harrowing to see for me. We've done some gene expression studies where instead of just handing the placenta off, we biopsy it, we do transcriptional profiling. And again, this is a different study. We see pretty profound differences in gene expression patterns by maternal SES and the placenta. These are all biopsies of the chorionic villi, again, that layer where nutrient exchange occurs. And we see this pretty shocking pattern that there's a pretty consistent upregulation of transcription factors that are involved in inflammation, B-cell activation, T-cell activation, macrophage activation of the placenta that varies with SES. And sometimes the differences are somewhere between 1.5 and 2.5 statefold increased activation of these transcriptional factors. So what we see is maternal disadvantage associated with more placental inflammation, both at kind of the pathology level, where they're staining using histologic procedures, but also it shows up in the gene expression profile, and we get some clues to what the cell types and what the transcriptional pathways are going to be. At the same time, we see that increased expression of inflammatory gene networks. We see decreased expression of gene networks that are involved in fetal organ development. So these are the placenta, these aren't in the fetus, so we're not looking directly in what's happening in the developing fetus. But what we do see is the transcription factors that we know are involved in brain development and in cardiac development and in immune development are all consistently downregulated in the placentas of disadvantaged moms relative to the placentas of more privileged moms. It's a fairly small study, it's 110 or something like that. It's not huge, but these studies, as you can imagine, are easy to do. Samples are hard to come by. The profiling is not expensive. We're trying to replicate it. But these findings suggest to us that what we're seeing in people's 20s in our earlier studies from the decades ago, the roots of those things are probably being established quite a bit early, and these data suggest in utero. So we're working hard on this kind of stuff to try and convince ourselves that it's happening in a more reliable way, and we're doing kind of larger and more rigorous studies to assess this stuff. But it happens early, and then we started to ask this question of intergenerational influences too. That slide I showed you about gestational age earlier, we're doing work on that in context with asthma too. So these are some data looking at a sample of 300 kids with asthma that we recruited in Chicago, which is another line of work we do. Hannah was very involved in this work, and still is. So we took the parents and interviewed the parents about their childhood socioeconomic circumstances, and then their kids are the ones who have asthma. These kids range in age between about 9 and 18. Drew blood from the kids, challenged their cells in vitro with a bunch of different ligands that mimic the kinds of things that trigger asthma. Cockroaches, dust mites, all those fun sorts of things. And then we look at production of cytokines that are involved in asthma pathogenesis. And you can see that cytokine production in these kids patterns by their parents' childhood SES, independent of the current family socioeconomic conditions. So the pattern is such that the kids whose parents had lower SES as children produce more of these Th1 and these Th2 cytokines, both of which ultimately result in the kind of chronic inflammatory processes in the airways that contribute to asthma problems. And then if we have parents and kids rate their asthma control, you see that things go the same way. And again, this is what's amazing about this to me, if it's true, is that it's independent of what's going on in the family socioeconomic conditions right now. This is all about what happened to the mother early in her life. So we're seeing lots of those sorts of associations, trying to figure out what they mean. I'm going to take a quick swig of water here. Now, of course, as I said earlier in the talk, the excess risk that is seen in epidemiologic studies is important. Understanding the mechanisms has some value, but it's only one part of this story, and perhaps not even as important as understanding the flip side, which is how some kids do well and who they are. And we have been working harder than ever on these sorts of questions about protective factors and resilience. There doesn't seem to be at this point in the United States the political will or ability to make large-scale investments in our economic infrastructure or in redistribution or any of those dirty words. So if we can't change the broader socioeconomic context through policy at this point, at least at that level, then trying to understand what kinds of strengths we can find in children and families and neighborhoods and communities and parlay those into interventions and changes. So one of the things that we've been focused on, of course, in thinking about this, what the protective factors might be, looking at the developmental psychopathology literature, the criminology literature, the educational outcomes literature, and the protective factors that one should focus on have been really clearly and powerfully identified by investigators going back decades, way before I was thinking about this stuff. And so their work has just been this amazing stepping stone for us to look at. So the obvious thing to look at with low-income kids who are at risk, what would that literature tell you to look at? Well, you should look at caregiving, right? That there's an amazing literature dating at least back to M.B. Werner's classic studies in Hawaii, on the island of Kauai, looking at mentors and caregivers and nurturing adults. So that was the first thing we looked at as a potential protective factor. We also have done quite a bit of work on self-regulation and self-control. Again, another protective factor that's been widely studied in those other literatures. And we see, at least with maternal nurturance and caregiving more generally, the kinds of buffering effects that all of you would be unsurprised by, but hopefully pleased with, because they're a big cause for optimism, and it's nice to see that they replicate across systems. So, here's some early work we did where we just went and used some of the data I presented at the beginning of the talk on early life SES and crossed early life SES with people's retrospective reports of maternal warmth. And of course, this design has all sorts of issues with what those retrospective reports mean. We don't think for a second that they're veridical, but we also think that they're telling us something about their early experience. And what we see in these very rudimentary, hard to interpret sorts of findings is a pattern that's pretty surprising given how crude they are, which is that we just focus on, these are young adults in their 20s and 30s, and if we just focus on the ones who come from lower SES backgrounds, we see that those who report, I came from a low SES background as a child, but I also simultaneously reported high levels of maternal warmth. Their gene expression profiles look just like the kids in our sample who came from more privileged backgrounds. So we see evidence that's very consistent, although not establishing this kind of buffering effect, right? So we see that in these sorts of analyses, we've done analyses in other data sets to look at the same sorts of questions. So these are findings from the MIDAS study. This is midlife in the U.S. Carol Riff at Wisconsin is the PI. This is about 1,200 midlife adults. We're looking at metabolic syndrome risk as the outcome, according to childhood socioeconomic conditions and retrospective reports of maternal nurturance. And what you see here is that metabolic risk at midlife patterns by childhood SES, independent of other risk factors for metabolic risk, but it patterns most strongly and most steeply in those who simultaneously report having received low levels of maternal nurturance and warmth during childhood. In fact, there is no gradient statistically for those who retrospectively report high levels of maternal nurturance. Again, these data aren't perfect. You've got retrospective reporting problems. You don't know how critical it is. We don't know what it means. But again, consistent with the idea that nurturant caregiving can potentially offset some of the health risks and biological risks that we've seen in other studies associated with early life socioeconomic disadvantage, and also paralleling again those other literatures on academic outcomes, we got tired of kind of second guessing what these observational findings meant, and thought we really need to do some intervention work to see if these findings are what we think they are, and if they have any relevance. So we were lucky to, at that point, start a collaboration with Gene Brody at the University of Georgia, who has since become a really close friend and collaborator. And Gene's done these amazing studies of low-income youth in small rural communities in Georgia. And just to give you a sense, in these studies, as I said, Gene uses this family-oriented intervention that I'll tell you more about in a minute, with African Americans from small rural towns in the South. And the intervention is focused on 11-year-olds as they're about to make the transition into middle school, not medical school. And a caregiver, typically a mother. And the intervention was an 8-week, once a week, 3-hour session with the first hour. For the first hour, the parents went in one subgroup and the kids went in another subgroup. The kids focused on things like setting goals, resisting peer pressure, building better social support, dealing with race-related stressors and developmentally related stressors around substance abuse, discrimination. The parenting side of things happened at the same time, and the parents in these sessions really worked on what Gene calls vigilant, involved, nurturant parenting. I think there's another modifier in there. He has a tough time getting into these words, and I think that's because it's a much more nuanced and complex form of parenting than many of us perform or know of in academic settings. So, the intervention program, I think, really deals, quite frankly, with the realities of what it's like to be growing up as an African American young person in a small rural community, and what the challenges are going to be. And it's pretty head-on with the parents about what the challenges are going to be, but also how to work with their children, how to be involved, how to be vigilant for problems, how to intervene, and at the same time, how to give your kids the autonomy and space they need to develop into adolescents and adults. And then the last hour of each session is all about communication and problem solving, so the parents and the children need to facilitate it. So this all happened as part of a NIDA-funded trial on substance abuse prevention that Gene and his colleagues did when the kids were 11. At age 19, we got involved and said, can we go draw blood from these kids and study inflammation? And we managed to pull it off. Gene's group managed to pull it off. So this will give you a sense for the sample. So this is a reasonably disadvantaged sample, at least from the kind of health psychology perspective I come from. So you can see about half the sample below the poverty line. You can see that 2/3 of the house is single parent caregiver. 2/3 of the family's parents have a high school education, but less than 10% have college. And at age 19, again, we drew blood. We measured 6 different inflammatory biomarkers in these kids' blood, and we made a composite because they all land on one factor and principal components. And what you see is that 8 years later after the intervention, there are pretty striking differences in inflammation between the kids who were in this intervention, called strong African American families, and controls who were not in the intervention. And, the effect size here is about 2/3 of a standard deviation 8 years later. So suggesting that Gene's intervention, potentially really changed inflammatory trajectories in these kids. And then you can do the path analysis to look at how much of that intervention and associated change in cytokines is mediated through changes in parenting. So they measure parenting quite well and quite thoroughly before and after the trial. You can create a weight difference score. And in the path analysis, what you find is that some of the association between the intervention and inflammation goes through changes in parenting. Not all of it does. In fact, more of it doesn't than does. But this indirect pathway is statistically significant, providing some support for the idea that by changing parenting and changing caregiving, you potentially may offset So these findings were inspiring and happy for us. Am I pretty much done now? I can't remember exactly how much time I have. 5 minutes. You want me to talk or answer questions?
I'll have some questions after.
Okay. So we've done, so we're working on this kind of stuff. You know, this isn't the perfect study because they didn't set out to measure cytokines at the beginning of the study or measure inflammation. It was a drug abuse trial. Ideally, you do this prospectively, and we're trying to convince, you know, the gods that be to fund us to do such a thing. We'll see if it can happen. It's going to take a long time to collect these kind of data all over again and mobilize the resources. But promising preliminary findings, consistent with the idea that caregiving might have an offsetting, potentially causally offsetting or buffering effect on these outcomes. We just don't know. We've also done a lot of work on self-regulation, and here the story is has been much more sobering and much more difficult. And we talked about this over lunch with graduate students today. We sort of assumed that in the same way that when you're looking at criminal justice system outcomes or academic outcomes or some outcomes related to mental health, that disadvantaged kids who nonetheless had high levels of self-regulation, perseverance, self-control would be buffered from some of the risks associated with low SES, that would also be true in terms of physical health. And we actually find quite the opposite. So just like other studies, what we find in Jean's work is that if you draw trajectories of kids' psychosocial functioning across late adolescence, so these are studies from 17 to 20, where every year we measure substance use, we measure depression, we measure aggression, we measure internalizing behavior. And then we can use latent class analyses to, I'm sorry about the size of these, but to divide the kids in the studies into groups based on SES and self-control. Right, so think of this as a 2x2 matrix. All these outcomes pattern not by SES, they pattern by self-control, right? Psychosocial. The kids who have good self-control have less substance use, fewer depressive symptoms, less aggressive behavior, all the good psychosocial outcomes. This isn't novel. We're not doing anything here that 20 other people haven't done in the literature already. Self-control portends better outcomes for kids across the board, and in particular for kids from more disadvantaged circumstances. So we thought, great, you know, this is going to also be true for physical health, right? One goes with the other. Mental health and physical health can be put together, academic achievement and physical health. We've done 5 different studies of different biomarker outcomes or health outcomes, and it turned out not to be true. So this is just one where we focused on cellular aging in kids who were at age 22, and we did that by measuring DNA methylation patterns, and there are ways using DNA methylation patterns that you can estimate the age of a person's cell and compare it to the chronological age, and that discrepancy is supposed to be another measure similar to telomeres and other biomarkers of basically turbulence in the system and how prematurely tissue may have aged. And what we find, this is just one illustrative study, is that in the subjects in gene studies, things for epigenetic aging and other biomarkers like allostatic load go completely the opposite way. So here, if we're looking at the most disadvantaged kids in our sample, higher self-control is related to faster epigenetic aging. It's also related to higher allostatic load scores, more inflammation. Low self-control is associated with better health in the most disadvantaged kids. And the kids who are less disadvantaged, and all of the kids in Gene's sample are living in what would normally be considered economically challenging circumstances. But in the kids who are less disadvantaged, it goes the way you'd think, right? So more self-control is associated with lower epigenetic aging and less allostatic load and better cardiovascular health. So what we consistently find, much to our dismay and puzzlement, is that the most disadvantaged kids who are working the hardest, who are the most conscientious, who have the best executive control, have great psychosocial outcomes. They stay in school longer, they get better grades, they have less risk of psychiatric difficulties down the line, they have less risk of substance abuse problems, they stay in jobs, they provide for their families. But when you unzip them and look at health indicators, they look much worse than their peers. In fact, in these studies, we've looked at kids who do and don't go to college from the rural communities in Georgia. And what we find is actually that if you just take the really disadvantaged kids and compare the ones who do and don't go to college, the ones who go to college have allostatic load scores that are like twice the level of the kids who don't go to college. So we keep finding this pattern, and again, we need to do more work on it to figure out what's going on and how it is. But it suggests that, conceiving of resilience in this simplistic unidimensional way, which is how we've always thought about it, either you're a resilient kid or you're not, is probably way too simplistic. That there are a lot of different layers to this, and that protective factors for one set of competencies and outcomes aren't necessarily going to be protective factors in other domains. It's reminded us of how tough this work is to do. So that's about all I have to say for now. This is all work that's highly collaborative in nature, and I never could pull any of it off without having great colleagues. Of course, I get all the blame, so if you have problems, talk to me. Credit goes to all my good collaborators and friends and the fantastic people we have working with us in Evanston. So I just want to say thanks very much, and you've been overly polite, so feel free to talk to me now and ask questions.
Thank you. That was a great talk. I think I've learned that self-control is terrible for you. I was curious if you could say more about the allostatic load. What were there particular components of the allostatic load? Which allostatic load?
I hate allostatic load.
Yeah.
That's why I don't present it, because I just hate it. I trained in Pittsburgh. If you even say allostatic load, they kick you out of town. But this was a great example of an allostatic load. So in the previous studies, it's a pretty simple measure looking at body mass index, resting systolic and diastolic blood pressure, C-reactive protein. I think there was fasting glucose in there. So it's more of a cardiometabolic risk profile. There's also urinary overnight catecholamines and cortisol in there, which makes it more like an allostatic load measure than a simple cardiometabolic risk.
Did you recall which of those components was contributing to?
Yeah, so when we break it apart, in that particular study, the catecholamines are probably the strongest component of it. But we've done work in ad health and in other cohorts where we see the effects more consistently coming out for glucose regulation, insulin, and more metabolic stuff. So I don't think any one component is driving it. What I do think is that different kids get there different ways. I think it's not a single pathway. I think for some of the kids, and we started exploring this in a little more detail lately, that different kids may cope with the challenges that we're seeing by using drugs, some may cope with eating, so we think that there are probably multiple pathways to which you can get to that cardiovascular health in these sorts of contexts.
So you wouldn't put this allostatic load idea towards the epigenetic as mobilizing resources, say through sympathetic activation, causing a little more wear and tear, or?
It could be. It could be that there's, so Sherman James has talked about John Henryism, which is this concept of very effortful, vigorous coping that he's identified groups of predominantly African American, although not fully African American samples lately. So it could be, and we certainly have anecdotal evidence for that, that it's this sort of hard driving, I have to succeed because my family and my community have sacrificed so much for me to succeed. So these kids do tell you, I go to college and I work all the time, I don't do physical activity, I don't sleep because I'm always studying, I gotta succeed. So there's a sleep story potentially there that we haven't been able to really get at. There's just a hard driving, I'm always aroused and going story there too. But there's also, I think, just a general not caring for the self in terms of health because other things are taking priority that might cross all that stuff.
Can you talk a little bit about your measures of socioeconomic disadvantage? You started with labor, non-laborers. We talked a little bit about high school educational level. If I had to pick one, or if I had to pick a couple, what would be your best guess about the relevance?
What matters? Is it resources or is it education, prestige, resources, those things? It's a good question. In some ways, a lot of these studies, both ours and the larger epidemiologic ones, have been opportunistic, going with what existing cohorts we have, as opposed to doing the prospective studies where you could more carefully characterize that. So, I don't think we have an empirical basis yet for saying, if you're going to put them in a horse race, which one would you go after? Conceptually, you can certainly think about different aspects of socioeconomic status as working through different pathways towards these outcomes. You know, in our own work in Chicago, and we just moved to Chicago 4 years ago. We moved from Canada, where the socioeconomic situation is very complicated. But in Chicago, it does seem to be a resources issue. We just see really tons of range in what kinds of economic and community resources families have access to, and that really is the differentiator. Education and occupational prestige are mattering quite a bit less in Chicago than they did in Vancouver, where we were for 10 years. I think because there's just a much stronger economic safety net built into the Canadian system. So, you know, our experience is that it's a little bit variable depending on where we're looking. We don't find much action with the more subjective aspects of SES, which is, you know, what a lot of psychologists have been interested in, and you know, good friends of ours have done a lot of that work. My own hunch is that the latter and those sorts of subjective things probably are pretty important in explaining variations kind of mid to upper end of the socioeconomic spectrum, and that's why, you know, with health you do see a fairly graded association, so that even upper middle class people's health is worse than truly upper class people's, and there I can see how subjective SES really could play out. But we're often looking at the lower end or low compared to high, and I just don't think that's what it's out. But if you have thoughts, I'm always eager to hear. We're always trying to do better on the socioeconomic side and on the biological side. You know, you get into these interdisciplinary studies, and it's never quite adequate in any one domain. Let's see, who is next?
So for some of the results you presented using the Strengthening African Families group, I know they collected genetic information, candidate genes, and they published the differential effects of their intervention. So I was curious if you tested for those or controlled them in your findings.
Yeah, it's a good question. Yeah, they've done serotonin transporter stuff and glucose receptor. You know, we don't see anything with those either as effect moderators or immune effects and the kinds of things we're looking at, but we're not that well powered. You know, once you get down to the number of people, you know, especially when you've got one under allele frequency, you've got only some sample amount of biomarkers. So I wouldn't say that the jury is out on those things, but you know, we don't see anything that convinces us it's going to be really important.
You're in Chicago now, where the city is very well mapped.
Yep.
Have you been playing at all with neighborhoods?
Yes, much more. So, you know, this asthma study that I briefly presented is our first data collection in Chicago. We've been there almost 5 years now, so we've got extensive neighborhood data, and we're just starting to analyze it and look at those sorts of questions.
Are you using traditional measures for neighborhood or some of the newer like activity space? We're collecting some in Philadelphia. We have traditional measures and then we have activity space research.
Yeah, so we're doing a little bit of both. I mean, this is a new area. It's certainly not our own traditional area of expertise. So some, you know, social explorer-based measures of kind of broader census-type data, and then we're also doing using a lot of Canvas Odgers approaches, where we go in and using, you know, Google Street View, coding aspects of the neighborhoods, and we're trying to make connections with the Samson folks to see if they're willing to share data too. So we're just getting into this, but I think in a few years with the right collaborators, we should be able to make some headway. I mean, it would be great to be able to connect the real contextual stuff that sociologists have done so well with, you know, the biology that I think, you know, we can do, you know, on a smaller scale, but it couldn't be done there. So it's happening. It's just slower than you'd like. That's a great question.
I think we'll have to have questions there. I just do want to make a very quick announcement that we did record the session. I think we were supposed to say that at the beginning, but anyway, the session's recorded. If anyone has any concern about that, they can contact me. And I'd like to thank Dr. Gregory Miller again for giving a lot of his time to us. He's coming for pretty well a 2-day visit here. Most speakers come for a day, which is fantastic. And I'll just ask the BBH grad students to please stick around for a few minutes. Thank you.